I want to welcome everyone here who's here in the classroom today, as well as those who are watching via Zoom. I'm Tom Wolf, and I'm a member of the HASP Science, Medicine, and Technology Committee, and I see one of my fellow committee members here. I'm here to introduce our speaker, Dr. Naman Vasta. Before doing so, however, I want to acknowledge the contribution of the Van Andel Institute, letting him appear today. That's very generous of them. We greatly appreciate their support. Dr. Vasta is not the first VAI researcher to speak to HASP. In fact, he's the second. A year ago, there was a presentation about Parkinson's disease, which was, again, like this presentation, very well attended. Dr. Vasta earned a PhD in neuroscience from the National Brain Research Center in India. During his PhD studies, he worked on several projects ranging from basic to translational studies on Alzheimer's disease and Huntington's disease. Since June 2021, Dr. Vasta has served as a postdoctoral fellow at the VAI, where he investigates the tau protein, the misfolded protein in Alzheimer's, and he also studies the underlying pathogenesis of Parkinson's disease. His presentation today is entitled, "What the Latest Research on Alzheimer's Tells Us." And I'd ask everybody to hold your questions, please, till after he completes his presentation. So, Dr. Vasta, the floor is yours.
I hope I'm audible to everyone. Yes, okay. Thanks, Tom, for the introduction and for this invitation. I'm very glad to be here, and I hope I serve the purpose and solve some of your queries which you have. So moving ahead, at Van Andel, this is the group I work with, and last year, my supervisor, Mike Henderson, gave the presentation on Parkinson's, and we all work towards a common goal of understanding neurodegenerative disorders and how we can leverage the research opportunities to find a cure to slow the progression or stop the disease. I have laid out a QR code which leads to the lab website. You can scan it, and as well, you will land up to a page which talks about what kind of research we do, and we timely update this on the research we are doing. We can also look at the Twitter accounts, mine, the lab, and the Van Andel Institute to get the updates of what kind of research is being done. Okay. In today's presentation, I'll talk about starting with brain, because to understand a disorder, we need to understand the organ, how it functions, and what happens so that a dysfunction leads to a disorder, which leads to several neurodegenerative disorders. I'll focus on Alzheimer's disease today. Possible causes, what leads to Alzheimer's, what are the therapeutic avenues researchers are looking, what are there at the current clinics, ongoing research in terms of understanding the disease, and I'll take any questions which you have. So brain, it's a very, very complex and fascinating organ. It's composed of billions of neurons, which is the key cell which makes the brain. It's also composed of other cell types like microglia or astrocytes. This is the neuron, how it looks like. Each of these cell types have different functions. Like neurons are the key function, which functions of however we behave and work, these are the ones responsible. Other cells are the supporting cell types. For example, the microglia, they are the debris clearer of the brain. So whatever byproduct, waste products comes into the brain, it helps to get rid of it. Astrocytes support nourishment and nutrition to the neurons. So the primary agenda here is to make neurons function by the supporting cells. Each region of the brain has different function. So unlike other organs like heart, liver, which does one function, for any other function, brain is the responsible organ. So each region, it looks like a homogeneous brain which similar architecture, but each of these region has a different function. For example, this region is called, just to clarify the terminology here, the one fine line you see, which is a dip in the brain, it's called a sulcus. And the bulges are called the gyri. So this sulcus is called the central sulcus. Ahead of this lies a region which we called as motor cortex. This motor cortex is responsible for all the movement we do, any kind of motor movement. It's also segmented, like a motor cortex right here is responsible for our facial movements. Motor cortex right over here is responsible for our trunk movement. Motor cortex over here is responsible for our limbs movement. Right ahead of it is a premotor cortex. What it does all the planning before we move or execute any function. For example, if I plan to pick this bottle up, before even I initiate this movement, my premotor cortex has planned how much my palm should wide open, how much length I have to go, and by what strength I have to grip it, and by what strength I have to lift it. Based on the assumptions, what my eyes have seen, that this is a bottle, it might have water, how much it weighs. So before I even move, my premotor cortex has planned everything and send instructions to the motor cortex to execute. Say, for example, I assumed this is a bottle full of water, but actually this is empty. How will I pull this? The moment I lift it like this, because my premotor cortex was confused about that, was not right on the weight of the bottle. Right behind the motor cortex is a sensory cortex, which does all the function, how we perceive the stimulus around us based on touch. And this is also segmented across this region based on the sensation we receive from limb to head. So this is the frontal lobe. Frontal lobe is basically addressing how my personality is. How do I talk? What are my planning things? Higher cognitive skills, thinking, planning, reasoning are governed from what is called frontal lobe. This is the occipital lobe, which is responsible for all our vision. Our eyes send signals to this region, and that's how we perceive the world around us. This is the cerebellum, which controls the balance and movement. Here is a region called medulla oblongata. This is the most vital region of the brain, which controls our breathing and heart rate. What I mean to say here is all different brain regions have very different functions, and they control a very vital part of our being. If we move and see the inside of the brain, we see different structures here in the midbrain. One key structure here is the hippocampus. It's shape of a C-shape. Hippocampus means seahorse. People who named it kind of see that as a resemblance of a seahorse. They named it hippocampus. This structure is also very interesting, important. And this is the structure why we see what happens in Alzheimer's. This structure is responsible for new memory formation. Once the memory is formed, and over the period of years, it goes from hippocampus and stores elsewhere in the brain. But the new memory always starts formation in the hippocampus. There's an interesting story how we learned about hippocampus, and it's a short-term memory. There was a patient called HM who had epileptic seizures back in the 1950s. He was diagnosed that the epileptic locus is in the hippocampus, and later the surgeons removed the epileptic locus and hippocampus was removed. Well, the seizure was cured, but HM did not make any fresh memories. He had all the old memories, but he could not make any new memories. And that's when people realized how important this structure was. Another region you see here, right beside hippocampus is the amygdala. This is the site of all the emotions we generate: love, fear, hate, anything. All the emotions are governed from this region. I'll explain to you, and all these regions are connected to each other like a mesh of network of neurons. They have to communicate within these structures for us to function. I'll explain how this connectivity is important with one disorder. It's called Capgras delusion. It's an ultra rare disorder. What happens in this disorder is the connection between visual cortex, what we see, and here is a region called fusiform cortex. Fusiform cortex is responsible for facial recognition. So if I identify you by face, that's because my fusiform cortex has registered that face. Now visual cortex, because I see face, sends signal to fusiform cortex, recognizes face, and fusiform cortex sends signal to amygdala, because if I'm familiar with you, I have certain emotions for you. Now, due to some accident or some other reason, if this connection is lost from fusiform cortex to amygdala, the patient will see a face. For example, a patient is there and his or her mom comes in, he would say, oh, he recognizes the face because his visual cortex is fine. He could see, he could identify and say, you look like my mom, but you're not my mom, because he doesn't generate emotions seeing that face. But if the mom goes to the next room, gives him a phone call, he say, hey, he would say, hey, mom, come to this room, because his ears to amygdala connection is still intact. So these neurons have to communicate for us to function. So a loss in one region is the not loss of that particular function. It's a loss of all other functions as well. And how do they communicate? One neuron to other neuron communicates through chemical messengers, which in turn leads to electrical activity. This connection between one neuron to other neuron is termed as synapses. And there are billions of synapses. We make synapses, neurons connect, they disconnect, and that's how we form memory. One peculiar characteristic of neurons is they don't divide. There are certain, latest research has shown there are certain regions where you do certain new neurons are formed, but that's minimal. Mostly neurons don't divide, whatever we are born with the same number of neurons. Now, what happens in neurodegenerative diseases? This is a healthy brain. Just like our house, our brain functions, there are byproducts, there are waste products, and there are people, us, who would take out that waste and throw it and recycle it. Similarly, in the brain, that's recycling happens. Byproducts are there and they are thrown away. We excrete those things. What happens in a diseased brain? The messengers, the proteins responsible for clearing this debris are not that efficient. So they do clear, but not enough. It's still there. And over a period of years, when we age with a defected protein clearing machinery, leads to several neurodegenerative disorders where brain degenerate. So here we would not see any kind of symptoms because the machinery is still functioning. Cells are still compensating for this waste byproducts. It's still now very hard to know people, like diagnose people at this stage. We only know when there are symptoms arise. Now, there are several types of neurodegenerative disorders. These disorders are classified based on what are the toxic proteins which get into the brain. For example, TDP43, A beta, that's for Alzheimer's. tau for Alzheimer's, synuclein for Parkinson's. So this is an ALS, amyotrophic lateral sclerosis, frontotemporal dementia, corticobasal degeneration, progressive supranuclear palsy, Alzheimer's, dementia with Lewy body, Parkinson's disease dementia, and Parkinson's. If we see classical TDP-43 and sum of tau leads to ALS and FTD, tau leads to several other disorders. Synuclein leads to Parkinson's, A-beta leads to Alzheimer's and Parkinson's. It's a combination of these toxic proteins accumulation what leads to different types of disorder. Another factor which plays into the role is what brain regions are involved. If hippocampus is involved, it leads to Alzheimer's disease. If the initiation site is the basal ganglia or the substantia nigra over there, leads to Parkinson's disease. If striatum is involved, it leads to Huntington's disease. If in the spinal cord, motor neurons are involved, leads to ALS. So it depends on what kind of toxic protein is there and what brain region gets affected by this, decides what kind of disorder it's going to lead to. Now, Alzheimer's, coming to Alzheimer's, I'll state some facts which are really disheartening, that it's the most common neurodegenerative disorders. Currently, as per the CDC, there are 6 million patients in US, and with the current rate of increasing patients, in 2050, they predict we'll have 13 million population suffering with AD. Since 2019, the death rate has doubled. It's even surpassed HIV and other infectious diseases. And strangely, it affects more so women than men. We don't know. That's opposite of what we see in Parkinson's. Parkinson's male are more susceptible for the reasons we don't know. And today, to support Alzheimer's care, it's a $321 billion expense to the US economy, which is a lot. Now, what happens in Alzheimer's disease? First of all, it initiates in the hippocampus, starting with hippocampal dysfunction. And what hippocampal does is make fresh memories. So we have hindrance in making fresh memories when Alzheimer kicks in. It's also a progressive disorder. So it does not just stay in hippocampus. Over a period of decades, it involves other brain regions. And at the same time, what brain regions are involved, we see dysfunction of those brain regions and which are reflected as a symptom of this disease. And that's right. Now, what are the stages? The mild stage is like very little symptoms, hard to detect, hard to even see that, whether it's like memory loss of recent events. Even a common man, even without AD, would experience sometimes difficulty in organizing and expressing thoughts, getting lost or misplacing belongings. Even a person without AD would have these symptoms. Not necessarily it leads to Alzheimer's, but people who have later in life diagnosed with Alzheimer's have experienced this at the starting point. If the disease progresses, they experience more severe symptoms, and at the later severe condition, they'll have round-the-clock assistance with daily personal care, loss of awareness of recent experience, have difficulty in communicating. I would just like to clarify, not all patients diagnosed with AD would lead to a severe AD. We don't know who will and who will not, but not everybody would lead to from mild to moderate or severe. Some people with AD would always stay under mild conditions. But it's a very heterogeneous kind of symptoms we see in AD patients. We call dementia, that's the most common symptom we see in AD patients. But if a patient has dementia, that not necessarily means that the patient has AD. It could be a vascular dementia, it could be a Lewy body dementia, it could be frontotemporal dementia. But there are high chances that could be AD because under the umbrella of dementia, Alzheimer's is the most common reason. But one should not be confused to directly correlate Alzheimer's with dementia or dementia with Alzheimer's. Now I talk about in the classification of what neurodegenerative disorders are classified into. Alzheimer's disease had two core pathological proteins. One is called the A beta plaques. A beta plaques are formed in between the neurons, and tau tangles are formed inside the neurons. This is an image of tau tangles, how they look like in a postmortem brain. And this is how an Alzheimer plaque look like in a postmortem brain. How we do this is through using very specific markers against A beta and tau tangles and stain these brain sections using a technique called immunohistochemistry. Now, what's happening in a healthy brain? Both A beta and tau are present in a healthy brain. They have normal functions. For example, tau supports the entire cytoskeleton of the brain, the neurons. A beta controls how neurons fire through potassium channels. But under a disease condition, A beta gets to accumulate it in terms of plaque and creates a waste burden for that neuron. This waste burden causes the neuron to dysfunction. And this dysfunction further leads to formation of tau protein tangles. And eventually, neuron loses its functionality and they die. But A beta and tau are not the only reason of what we see as a symptom in Alzheimer's. They might be the starting point, but if it leads to vascular system. Vascular system, when it gets damaged, the brain doesn't receive enough blood, nutrients for it to function. Leads to a generalized inflammation inside the brain. It hinders the metabolism. There's not enough glucose, glycogen going to the brain for enough energy to function. And there is oxidative stress. Free radicals are generated as a byproduct of our metabolism of ATP generation, which is the energy molecule for our biological cell. And all these things together lead to what we see as an AD symptom. Now, what could be the risk factors? There is no one cause which leads to AD. There is no one risk factor which could lead to AD. It's a combination of all of them together which leads to AD. Age is not the cause of Alzheimer's. There's healthy aging, which is there in a lot of people, but it is one of the risk factors and the most common risk factor. Genetics. Some people are born with certain genes which make them prone to Alzheimer's. Some people are born with genetics which make them prone to Alzheimer's, not necessarily means they will lead to Alzheimer's, because they might not have the other factors like metabolic syndromes like cardiovascular syndrome, diabetes, obesity. Physical activities like what kind of diet we are taking, smoking, drinking. Family history. Does our family members have Alzheimer's or not? So even if there is just one family history, you have a genetic predisposition, still, you might not lead to Alzheimer's because you don't have other risk factors. And there are others like education. Research has shown that if people who are involved in education, whose brain are cognitively active into planning things, doing things which involve reasoning, they exert their brain to do some tasks. They are much less prone or susceptible to have Alzheimer's. Now, biomarkers as disease progression. Now, research community, we want to know, first of all, say, whether this is Alzheimer's or not. or what stage of Alzheimer's this is. For that, research community is developing biomarkers. For example, this is a classical figure. What you see here is the disease age. The red line is A beta. The blue one is the tau. And what you can see here, it's written cognitively normal. So even when there is a buildup of A beta, at a later stage buildup of tau, still in this window, the person can remain normal. It's here in a very short window when the A beta peaks up, experiences mild cognitive dementia, NCI. So these markers can serve as a biomarker to predict at what disease stage the person is and kind of predict whether this person is likely to go to AD condition even before the symptom comes in. How we diagnose Alzheimer's, how clinicians, maybe a clinician would be a better person to say, but they assess based on patient history and physical exam. They do some physical exam, then reflex tests, assess patient's medical history, what are the metabolic syndromes, the family history, the genetics behind it. They also do some behavioral tests. Recently, FDA also approved some computerized test, which doesn't involve just asking questions. It's a computerized test to assess your ability, cognitive ability. It's a brain imaging. We do MRI, structural MRI, to see how brain has atrophied, see hippocampus, if it's normally looking or not. Recently, cerebrospinal fluids, which is taken from the spine at the lower lumbar region to assess some biomarkers. And of lately, research is also interested in blood tests because blood tests are more convenient to take. It can cater larger population in a community than a brain imaging or a CSF test, which requires specialized center to do that. Although these are not routinely practiced because still much of research has to be done to say with certainty that this would lead to a meaningful assessment. So it's an amalgamation of all these tests when a clinician can conclude whether you have Alzheimer's or not. No one test is a sure shot test to say whether you have Alzheimer's. Now, why biomarkers are necessary? It would if we identify AD, it would differentiate AD from other dementias and would help decide what kind of medicines the patient would need. We will identify what disease stage the patient is, and alongside we can address with the medications. Identify appropriate people for clinical trial. Every person is different. Every person is at a different stage of disease. One drug does not fit everybody. So we have to identify what patient population would be beneficial for this particular drug. And biomarkers would be helpful identifying those people so that every population gets a different set of treatment which works for them. And if we are able to succeed in identifying these biomarkers, we'll be able to diagnose AD 20 years before the first symptom comes, and we'll be able to help them and treat them before even they go to Alzheimer's stage, and they don't even go there, and we kind of stop the progression and make healthy aging possible. Recently, there are two tau isoforms. These are phospho-tau 217 and 181, which have gained a lot of interest because these ones are the ones which are identified at a higher percentage in Alzheimer's CSF and blood tests. Likewise, there are many other markers which the research community is addressing to identify, validate among huge population to say without more certainty what could be the best biomarker to assess. So what challenges as a researcher we have for this disorder. First of all, it's a progressive disorder. It keeps on becoming severe over a period of time. And it's a very slow progressive disorder. It takes decades for a symptom to come and go to a next stage. So if you invent or discover a drug day one, the effects of the drug, whether the disease has worsened or you have stopped the disease to worsen, it's a period of a decade to identify whether the drug has worked or not. The current evaluation methods are not that fine to assess within a short period of time. So biomarkers would help in identifying whether the drug has worked or not worked by identifying the biomarkers and not by visualizing or assessing the clinical signs or the symptoms. It's a very complex disorder. It is not a disorder that a gene is mutated and leads to Alzheimer's, and then you target that gene, work around it, and find a cure. It's a very complex disorder. A lot of factors come into picture. And that's why it requires multiple treatments. No one therapy would be useful for it. There would be multiple therapies which is required to combat AD. And we have, in the whole world, we have an aging population. Every year, we have increasing number of patients coming in. So we need to really pull the trigger and work in finding cure for this. How we do it? You'll have to diagnose Alzheimer's even before the symptom comes in. So we need to create early diagnostic tests. We need population-based studies. There is not huge representation from all communities who come to clinicians for them to identify, oh, how much population in this community has Alzheimer's. So we need a larger population-wide studies and person-specific studies so that treatments could be catered in an individual basis. Lifestyle interventions has a huge role in what leads to Alzheimer's or any neurological disorder. So these people have to be made aware how important lifestyle interventions are. And then address the disease pathway, what happens in the brain, what goes wrong in the brain, and then use those pathways to identify drugs. So, Alzheimer's disease was first identified by Dr. Alois Alzheimer back in 1906. It's been more than a century when this disorder was identified. We still are way behind of finding a cure for it, but research has progressed and have contributed a lot to the stage where we are right now. I'm just going to take you to that journey of important milestone, starting from 1906, how things have changed and what we have gained from research. It was named as Alzheimer's in 1910. In 1968, after like 50 years, we were able to devise an assessment tool of how we can judge a person has Alzheimer's or what stage of Alzheimer's they are in. More awareness was created. National Institute of Aging was founded in between. Beta amyloid was first identified in 1984, that this is one of the toxic proteins, Alzheimer's, which leads to Alzheimer's. Tau, very recently, now 1986, after that, was identified. APP, it's amyloid precursor protein. It is one of the candidate genes on which mutation have higher chances to lead to Alzheimer's, was identified in 1987. ApoE4 is one of the risk factor genes. If you have a mutation in this, you might have a risk of developing Alzheimer's, was identified in 1983. And first drug for Alzheimer's was approved in 1993. When the research progressed, we developed animal models, which was first in 1995. First MRI measurement of hippocampal atrophy was done in 1998. Memantine, which is still a drug of choice, was approved in 2003. PET scans to identify tau and A beta in patients who are living was done in 2004. First prevention trial targeting amyloid beta was launched in 2012. 2 years back, 2021, we FDA approved aducanumab for Alzheimer's. There's a huge debate going on that. And very recently, lecanemab was also approved for Alzheimer's. And again, a huge debate is going around that as well. I'll come to that in later slides. So what are researchers doing at Van Andel Institute for Neurodegenerative Disorders? We have a department called Department of Neurodegenerative Sciences. This is led by Dr. Darren Moore. And we work on several factors. For example, genetics. We want to identify genetic risk factors. What are the genes responsible directly for a disorder in neurodegeneration? We do that. We also study environmental effects like toxins in case of Parkinson's. We study epigenetics. Epigenetics, I'm not sure if you're familiar with that term, but genetics is the kind of DNA we have, and all the body cells have same DNA. But what makes a heart, the same cell turn into a heart, the same cell turn into a lung, the same cell turn into a neuron, is the epigenetics. What kind of genes would be expressed under what environment is the epigenetics. And environmental factors play a huge role in that. So we study how this epigenetic changes affect neurological disorders like Parkinson's and Alzheimer's. We do cell biology study. We identify different cellular pathways which are there in a healthy cell and in a disease cell. We also do neural circuits. Like I explained, each neuron is connected to other in a complex way to organism to function. We study neural circuits and how it goes bad leads to a neurological disorder. And in Parkinson's disease, the deep brain stimulation, where an electrode is implanted in the brain to stimulate a region called thalamus, which alleviates the symptoms of Parkinson's for a short while. It's a result of studying neural circuits. Once we have done all these things, we test these findings in animal modeling. We create animal models or find available animal models and test our hypothesis to validate it. If it passes that, we also go to clinical trial. So Patrick was with Van Andel previously, who initiated International Linked Clinical Trials Initiative. So far, this has been working towards Parkinson's and in collaboration with other organizations who can do clinical trials and help starting clinical trials, for example, in Broxol, simvastatin, exenatide, and liraglutide. We have West Michigan Neurodegenerative Mind Program. What this program does, it has created a biofluid repository, a brain repository, and an iPS circulation platform. But what are these? Biofluid repositories, we have a bank with biofluids like blood, and we identify different genetic variants in these populations. So we get blood from healthy people, diseased people, and we try to identify what are the other genetic risk factors in the disease population and document them to identify the particular risk factors in the population around us. We have the brain repository. We are very fortunate that we have approximately 100 brains donated by patients. We do histopathological analysis on them, and we provide clinicians a final neuropathological analysis from these brains that this was the histopathological analysis. This person in his brain had these many pathological proteins, and the clinician can correlate these findings with the patient history and come to a consensus which could be used for future patients. we have an iPSC platform. iPSC stands for induced pluripotent stem cells. So this came into picture because we were using a lot of mouse cells to study. Back in 2012, a scientist created iPSCs who took cells from the skin, made them stem cells, and then made back into any kind of cells. For here, makes a neuron. So they created neurons from skin cells, but it has the same genetic architecture of that person. And so for the brain repository, it's postmortem. This is from a living person. So you can study the genetics of that person and the cell biology using this platform. So Van Andel has also initiated this platform where we have a huge library of iPSCs from huge variety of patients and healthy volunteers as well. So what we do in Henderson Lab, the lab I'm associated with, we address how do these pathologies spread. As I told you, in case of Alzheimer's, it all starts in the hippocampus. And then as the disease progresses, it reaches different parts of the brain. We want to identify how this pathology spreads. And if we are able to stop it, we'll be able to stop the progression of the disease. We might not be able to stop the initiation, but definitely the progression. We want to identify the genetic risk factors with the brain and biofluid repositories. We also want to identify which cellular pathways are disrupted and target them so that the neurons function in a healthy way. And use all this to say whether we can stop it or slow the disease, at least slow the disease. Now, pathology is how they spread. This is a site where hippocampus is. Over a period of time, these pathological proteins move to different regions of the brain. We have developed animal models where we can study how and what circuit tau or A-beta follows from hippocampus to other brain regions. So we inject pathological protein in mouse in the hippocampus and study the trajectory. And in this due process, we have also identified that not all the cells in this trajectory would lead to tau pathology, for example. There are some set of neurons, even in presence of pathological protein, function exactly normally. Some set of neurons, even in the presence of tau pathology, will not develop tau pathology. And some are, so in general, some neurons are vulnerable to tau or Alzheimer's pathology, and some are not. What we want to do is to see the characteristics of these neurons who, even in presence of pathological proteins, are able to function normally. Understand what's their characteristics and use them, apply them on the vulnerable population of neurons. And in a way, we want to make the entire brain resist to these pathologies. So even when these pathologies are there, no neuron should dysfunction or die. That's the ultimate goal. Other thing which we are addressing is the pathology progression. So if from hippocampus it's going to different brain regions, we want to identify how it spreads, how one neuron takes up pathological protein and gives it to the other neuron. We want to understand specific mechanism behind it and then target it to stop this uptake and transfer it to other neurons so that it just, even when we identify the hippocampal degeneration, we stop it there. Also, when there is tau or A-beta plaque formation, so the neurons are dumping them to form them aggregates because these are misfolded waste product. Since the neurons are not functioning properly, in this process of dumping this waste product, some good proteins are also targeted and discarded, which leads to deficiency of these good proteins in the brain. So if we identify these good functioning proteins which are not misfolded, which are mistakenly discarded, and increase their efficiency, we might be able to alleviate some of the symptoms. So where are we in the treatment? How close are we there? What has the research done so far? In order to understand, let's understand the process of drug discovery, how that happens. Van Andel Institute is, what we do here is the drug discovery at the preclinical stage. What we do is target selection. Here, the target means what a drug should target at, what protein, what gene, what pathway. We need to identify them. Second, once we identify, we need to lead discoveries. We need to go through high throughput screening of the available chemical modulators to screen what are the drugs available, or we need to synthesize new drugs to target this. So we go through this high throughput screening in 96-well plate or 384-well plate. These are really sophisticated plates, and maybe 5 inches by 3 inches plate has 384 wells in them. Each well has neurons in them. And when you add drug, you can add 384 drugs at one time to assess them. If we find something out of here, we need to synthesize new drugs. That's part of collaboration. We do in vitro tests. We do several tests in a dish where we culture neurons in a dish, incubate them artificially in an incubator, and we test various cell biology pathway analysis. We see whether the drug is toxic or not, whether the drug is showing cell biological changes or not. And then these findings are then moved ahead in the in vivo animal model, because in vivo models are much more complex. This entire process takes close to 2 to 5 years if everything works perfectly. Once you have a lead from in vivo tests, we move to clinical trials. Clinical trials are designed in a way in 4 phases. Phase 1 is to assess the safety of the molecule in healthy people, whether this drug is safe. We have already, before reaching here, we have already proven that the drug is safe in culture, in vivo, and disease models. So we have higher chances of drug to be safe here as well. This takes around 1 to 2 years. We move to phase 2 here. Phase 2, we assess efficacy in the patient population in a smaller cohort. Then we move to phase 3, where we have a larger cohort, 300 to 3,000 patients, to confirm the findings of phase 2. Here, after success of phase 3, we move to FDA, submit data to them, and seek their approval. And then it goes to phase 4, when the drug is launched and patients in the general public are given those drugs and they are monitored closely. This entire process, 1 year, fastest 1, and then 2, 4, plus 1, 5. It'll take 5 years. So in general, right from the start of the hypothesis and the molecule which reaches a market, it's a journey of 10 to 15 years, which is a long, expensive, very exhaustive journey. So we also need to focus to shorten this for disorders which need ultra care. Like, for example, in COVID, all these processes were fast-tracked. All the strength were focused there to develop vaccines. So what we have as current therapeutic options, we have some symptomatic relief. We have some cholinesterase inhibitor, some glutamate regulators, and these 2 in combination. So choline, acetylcholine, is a neurotransmitter which helps neuron to communicate from one to another, which is disrupted in Alzheimer's. Glutamate is another neurotransmitter, and both these neurotransmitters are disrupted in Alzheimer's. Correcting them with these 2 drugs leads to some kind of symptom alleviation, like in cognitive symptom, memory, and thinking. But this doesn't stop the disease. There are other non-cognitive symptoms, like anxiety, depression, sleep disturbance. We have drugs to address this as well, so the patient can have a normal living with these drugs. We have 2 disease-modifying drugs. which was approved in 2021 and 2023 in January. Aducanumab and lecanemab. Both these drugs are antibodies which stop the formation of A beta plaques. Here you can see a comparison. These are healthy A beta, which leads to formation of plaques over here. One, lecanemab, has a higher binding affinity at way early stage, where it prevents the formation of these A beta fibrils. The other drug, aducanumab, has a binding affinity higher for the fibrils. So aducanumab helps in discarding these fibrils. Lecanemab prevents the formation of the fibrils. Lecanemab, the latest drug we have, it in a diseased brain. In a healthy brain, there are antibodies there, and in an Alzheimer brain, there are A beta plaques over there. Lecanemab binds to these Alzheimer plaques and prevent them to form fibrils. It also recruit immune cells like microglia to help this process. Now, this study, the phase 3, was done at a multi-center, multi-hospitals. Several hospitals were involved in it. It was double-blind. So the doctors didn't know which patient had the drug and which patient didn't have the drug. And the patient didn't know whether I have been given a placebo or I have been given a drug. So that's why it's called double-blind. It was randomized. There was no specific order of selection of the patients. It was randomized. Nobody knew what's going on. And a placebo control, obviously, there were a set of patients which were just given a fake drug, and they did not know that. So whatever, based on this characteristics, whatever data comes out, we have higher certainty of believing that data that this is true. This is a data published in the journal 2023. The green line is the patients which did receive the drug, and the other line is the patient which did not receive the drug. The drug was given for 18 months, and we could see reduction in the plaque load right after 6 months of the treatment, which the difference was way higher at the end of the 18 months, even though there was reduction just like in a patient population, but the process was slowed down. This is a picture, a complex picture, which shows what all drugs are still being investigated in clinical trials. This data is back in 2022. That's why lecanemab is still there in phase 3. So we have disease-modifying biologic. What I mean with biologic is these drugs needs biological system to manufacture them. They are not made in the labs. In the labs, not chemically, but biological system like cells or bacteria are used to make them. You have disease molecules, small molecules, that are chemical mediators. And we have some symptomatic and some disease-modifying therapy. Alzheimer's disease are way ahead, and there are a lot of drugs being investigated with the hope that there will be more drugs with positive results in upcoming decade. Right now, we have 143 unique therapies and 172 clinical trials being run. We have a total participation of 50,000 patients in these clinical trials. 9.8% of them are for cognitive enhancement. 6.9% of these drugs are for neuropsychiatric and behavioral symptoms. And 83.2, which is the most important, disease-modifying drugs to stop or slow the progression of the disease. Yeah. No, this is global. We have 31 agents which are at phase 3 clinical trials. So this would run for, at the max, 18 months to 24 months, and we'll have an outcome of phase 3 results, whether they're efficacious or not, in coming 2 years of these 31 agents. We have 82 agents in phase 2, which, if proven efficacious, can move to phase 3. We have phase 1, a lot of drugs testing for the safety in phase 1. 50% is funded by the pharma, 28% by the academic institutes and nonprofit organizations, 17% by public-private partnership, and 5% by others. So it's a whole lot effort from all the regions and all the funding agencies running to accelerate this process of drug development for Alzheimer's disease. In conclusion, I would like to say the brain is a wonderful and also very complex organ. It's a complex network of neurons, which leads to the function, how a person functions. When protein misfolds and cellular function are disturbed, circuit is disrupted, which leads to neurodegeneration. And scientists are still trying to discover how this process happens, how this function happens, in order to identify particular targets to stop or slow down the disease. With this, happy to answer any other questions. Yes. Yes. Oh, even Mike. Yes.
We have a friend who developed Alzheimer's when she was 53 and she just died at 59. And they told us it was early onset Alzheimer's. Can you maybe shortly address that?
Yeah, so if patient population having a lot of risk factors, specifically if there is a genetic predisposition, for example, the candidate gene of APP being mutated, or risk factors of risk factor genes like ApoE being there, the Alzheimer's disease onset comes way early. If you don't have a genetic predisposition, there are higher chances that the person will develop disease at a later stage, 65 or above. So genetic or familial history leads to early onset of the disease.
Thank you. We've all seen the ads about how wonderful these memory improvement pills are.
Yeah.
They all look like our grandparents. Now I'm a great grandparent, so I pay attention, but are they worth the investment, or do they really do any good?
Are you talking about the approved drugs or the symptomatic?
Prevagen, it would be the most common one. You've
Okay.
seen the ads, I'm sure.
So that, like I said, every person, every patient is different, and they have to be catered in that way. Right now, we don't have the capacity to test individual. It's like one treatment fits all. But it's true that these drugs addressing cholinergic and glutamatergic system, they do hamper these AD do hamper
Right.
these two systems, and we need to correct them. Not necessarily means these two are the only factors which leads to memory decline. But these two factors can be governed or regulated by these two drugs.
One other quick question. I think you mentioned in the beginning that genetics may be one of the most key factors in the whole
Yes.
problem. And we've read recently about changing one's genetics. Not quite sure how that's done, but might that be an ultimate solution to Alzheimer's?
Changing the genetics at that level would be like at the, yeah, so it's still at the research level and in the non-human subjects. Yeah, it's still not there. Very long way.
This is really just a curiosity question.
Yes.
I was stunned by the number of drugs in one of the 4 stages of clinical trial that you
Yes.
listed up there. Is there a rule of thumb of in any kind of clinical trials for any medication, a rule of thumb of how many of those that have one time been in one stage of the trial finally are proved to have enough efficacy to actually approve the drug and administer it?
So yeah, there are a set of rules. If I'm able to understand your question, you're asking what are the criteria to assess whether a drug A in phase 1 would move to phase 2 or
No.
Yeah.
Of all the drugs you listed up there.
Yeah.
How many of them typically graduate through phase 4?
most of them would fail.
You don't have a percent?
No. No, most of the hypotheses of drugs being working would fail, but the more we try, there are higher chances of one going ahead.
Okay.
The slide that you showed, where you were showing the results of the clinical trial,
Yes.
and you said it was a 27% rate of slowing for
Yes.
the treatment group versus the placebo group.
Yeah.
What was measured to show that it was slowing? What was the primary endpoint there?
Yeah, so these drugs are went to accelerated FDA approval. And this is the change from the baseline of the worsening of the symptom. And there is a particular scale to assess cognitive symptoms. And based on. Yes. Yeah. Yes.
So hypothetically, if you determine very early on that there's the disease and you remove the hippocampus, would that stop the progression?
Well, that's not the thing people would want, but yeah, but it's very hard to say, like, if person has, through biomarkers, if right now we say a person has A beta plaques or tau tangles, even though it doesn't have symptoms, that not necessarily means it would turn out to be AD. So yeah, there's no point of surgical intervention at that point of time. Yeah. Yes.
So once a neuron has been impacted,
Yes.
you can't fix it. There's no cure. You can only maybe stop the progression.
Yes.
Okay.
I'd like to ask a question.
Yes, I have a question. Maybe I didn't hear this correctly. I think you said that diseased brains cannot eliminate the brain's waste products or waste activity. More specifically, what are those waste products or things?
So in case of Alzheimer's, these would be A beta plaques and tau tangles. So we have a machinery in our cell and body, like one of them is called proteasome machinery. So they identify misfolded proteins, attach some tag to them so that other mediators can come in and degrade it. Now, where there is a lot of load of these toxic proteins and these machineries are overburdened and not being able to degrade these misfolded proteins, that's when these accumulation starts. Yes.
I have a number of questions there's 43 more participants online today. So here are some questions. If you could go to the funding slide.
Yeah.
There's a question about the role of the federal government, how much of the funding on the research is coming from the government.
Yeah. I think it's this. Yeah, this one.
There you go.
Yeah.
So 28% would include government and maybe some other non-governmental organizations grouped together.
Yes.
Okay.
This data was published 2022 by Alzheimer's Association, which is the organization which like leads this all this research.
Great, thank you. Next questions are around early identification. So would you recommend blood tests for biogenetic markers? And are there strong benefits to early identification at this point in time?
So blood tests are still at a research stage, still not there at the clinic. They are definitely beneficial if ever incorporated into the system, but considering not everybody, even though we do not have a very solid marker, you might have these biomarkers which we have studied and not develop AD eventually. Getting this tested right now would create anxiety.
Great, thank you. And then the last question is what about Pith's disease? Is that something related to this that you're aware of?
Yeah, Pick's disease, but yeah, I'm not familiar with the literature, like what the research and where it had
Different topic. Okay.
Yeah.
And then just a request to go back to the QR code that you had from one of your introductory slides. People
Yes.
interested in using that. And right now, that is everything from online. Thank you.
I have a question. You say that there is really no solid marker to look for in blood tests, so they're not useful generally to get some sort of detection about what early symptoms might be producing in the blood. But you mentioned also a CSF test. What does the CSF stand for, and what does it test for?
It stands for cerebrospinal fluid. It's the fluid which encompasses the entire brain and spinal cord, and the brain floats in that fluid. The exchange matter of toxic proteins or the drugs which goes to brain or comes out of brain happens in CSF apart from blood as well. This is an invasive procedure to take a sample from CSF, but it's much more reliable because it's direct contact with the brain.
So it would pick up some of those waste, maybe?
Yes.
Thank you. Just curious about the early slide you had about the increase in Alzheimer's versus or over time, and how it's
Yes.
supposed to be so much larger in a number of years. It sounds like it's faster than the population growth. Just a quick look at the numbers. So is there any correlation with other factors that research is coming up with why that is going up fast?
Because the world is going to be an aging population. That's what the trajectory is. So we'll have, since age being the major risk factor, so we'll have more of that. Still at the statistics level.
I know that the University of Minnesota is doing tests on dogs because they say dogs get the same diseases we do. And our dog had Bell's palsy, she had a stroke, blah, blah, blah. And so they said they're not using rats anymore because they have to put the disease into the rat in order to study it. And they're finding it's faster to find dogs who have these diseases. Have you all thought of doing that?
Well, the way research happens is like, we don't want to sacrifice these animals just for testing things. We go higher in the animal order only when we have solid evidences in early stages. For example, cell lines. For example, C. elegans worms or Drosophila flies. We start with these and try to gather as much information, because majority of the times hypotheses don't work. We thought this would work like this, but it doesn't in the biology. Biology is complex. So we want to address these hypotheses in lower order animals, try to avoid use of animals and address these questions and go in higher order only when we have to validate a very solid evidence. Yeah. Like, for example, high throughput screening, where we are screening 223 drugs, that cannot be done on dogs. You have to finalize at least 2 or 3 to do that. Yeah. Yeah.
My doctor has recommended a 4-hour examination at Mary Prebid.
Mhm.
And I'm asking it because it's not just my problem, but I suspect there are others in the room, because it took me 4 months to get on their calendar. Yeah. So there are a lot of people going through this. I'm curious if it's related to Van Andel. Are you getting the benefits of those examinations and findings and so forth? How do you coordinate all?
Not that I'm aware of. Not that I'm aware of. Yeah. But there is a huge patient burden, and as well as there are very less specialized center to cater patient needs at this point of time.
I think it needs to be said that you've taken an incredibly complex subject and made it almost understandable. And thank you for that.
We still don't understand it completely. I'm still exploring.
Looking at some of the medications in the past,
Yes.
they have, their efficacy has been about 6 months or so.
Mhm.
Has the length of time been extended on some of those medications, or are they more short-term?
So if we talk about the disease modifying drugs, which are claimed to slow the progression of disease. So first drug was approved in 2021. It was still a fast approval. There was a group of people who didn't agree with it because it didn't show good efficacy, but it did reduce the plaque load. So the FDA thought it was a safe drug. So let's give it out to people who are early in the disease. So if we reduce the plaque load way early at the disease stage, and as the disease progresses, in a decade, we would know whether it worked or not. So that, in keeping that hope, it was approved. So the phase 4, which is the post-market analysis, the drugs given to patients and then monitoring them because it's a safe drug, that would end close to 2030. So 2030 would be a year to evaluate the first approved disease-modifying drug, whether it was efficacious or not. The second drug did show effect in 18 months. The question is whether the effect stays like this. Do we still get improvement over time? That only time can tell. But these two drugs are IV infusion drugs. So these can only be administered at the special center. You go there, you have to IV infuse them. I don't exactly remember how frequent it is, but it is relatively frequent. So these drugs are new hopes, still not there, but yeah, these serve as the benchmarks to go ahead and prove that, oh, A-beta is the thing, tau is the thing, and we can work on more on it.
One last question.
Yeah.
Across the country, are there parts of the country that have more Alzheimer's diagnoses?
Yes, there is some data on that. I don't remember on top of my head, but there are pockets in the country where you see higher patient population, but there could be n number of reasons behind that, not just the factors more there. There might be people, more aware people who do go to see clinicians if they experience dementia. So that also could be a factor. So it's hard to rule out.
I thought maybe my question would piggyback on that as well. Would there be reason to think that more pollutants or different kinds of pollutants would be a factor in creating the increase? And
Well,
how are you finding that to figure in your research?
well, in the effect of environmental pollutants, it has a direct correlation with Parkinson's. Not that strong correlation for Alzheimer's. Yeah.
I have a history in my family of dementia. And I was hoping that with a blood test and finding out about the biomarkers, I would be able to know if that's a big risk for me, but apparently we can't know that yet. So would you speak to the pros and cons of knowing, and also what I should do? My mother had it. My sister died. She was 83 and she had dementia. So anyway, any ideas?
Yeah. So both these blood tests being the most convenient test for any disease for diagnosis, it's still at the research level for Alzheimer's to say that this particular marker is the one we should look for it. There's still more progress required to say with utmost certainties. These data are in small pockets, in small patient populations at 30 patient and 40 patient. That is not generalized for every other population in the world. So we are still in the research phase for these blood tests to say with utmost certainty that, oh, this is the marker you will have if you are going to have Alzheimer's in future.
Thank you.
I have a question. Is the Venando Institute involved in research on the continuum of care, translation?
Yes, so for Parkinson's, definitely, we have the International Linked Clinical Trials Initiative, which works with Cure Parkinson Organization, which is a British organization, to run clinical trials in UK and in US for Parkinson's. So on Parkinson's disease states, they have started this initiative of clinical trials. We don't conduct clinical trials at Van Andel, but in collaboration, we do work.
Anyone else?
Hello, I have a lot of Alzheimer's in my family. Also, my dad had it, my grandma had it, 3 of my aunts have had it. basically there really isn't I mean, other than eating right and exercising, there isn't anything specific that anyone can recommend not to get it, correct, is what you're, is what you're saying?
Yeah.
Okay, that's what I thought, but I just wanted to make sure.
You come, often you've said early detection. And when I wrote down the mild symptoms, I have a couple friends my age who are struggling through these things. Is when do we test? I mean, when do we come to a doctor and say, do I have Alzheimer's or am I suffering from dementia? I mean, have those first mild symptoms, I think you said memory loss of recent things, difficult organizing. These two friends were very good organizers at one time.
Yeah.
Is that, are these the kind of things when we should go to the doctor? We all think we lost something and we must have it.
That's what I said. While coming here, I lost my way. I parked my car somewhere else and I started walking here. So that's pretty normal. But if that is a pathological cognitive decline, a clinician would be a better person to tell. So that could be how aware our PCP are if we talk to them on these things and how good they are and how trained they are to assess these things. Yeah.
Yeah, I was having what I thought were some severe problems. I couldn't find Menards. I've been there 100 times. I couldn't find Menards and I couldn't find the Ford dealership. And so it seemed to be in the second or third stage of those stages you saw. So I went to my family physician and he's the one who recommended the Mary Free Bread place. So maybe that's the answer. I think you're having some problems beyond just not remembering where you put the car keys. I'm hoping that's going to prove to be helpful to me. I'll know on Wednesday, but
yeah.
I was just curious about talking about targets for molecular research. Are you aware of any effort to go after the cleaning system you described, where the bad proteins are cleared out of the brain, et cetera? Is there a way to support that system that offers any promise?
Yes. So not in phase 3, but definitely in phase 2 and phase 1, there are drugs targeting those protein clearing machinery pathway as well.
I believe you mentioned that there are some people who only have the mild symptoms and never progress through. And do we have any idea why that happens in some people and others not?
Not yet.
Not.
In any of the clinical trials, if Van Andel has a good idea and another company does, do you work together to maybe, oh, wait, here's an even better idea?
Yes. So yeah, that's up there for Parkinson's, but not yet there for Alzheimer's.
Do you know of the phase 3 trials, if there's any promising data on any of those candidates that are in phase 3? Or is it kind of like you don't know anything yet?
So when they have phase 3 done, results come out as a news and it's only get published and comes to the common public at a very later stage.
Yeah, no, I understand, but
Yeah.
sometimes there's like late breaking data or whatever,
Yeah, that could have commercial aspect to it as well. So let's not buy that. Yeah.
Is it reasonable to think that you still use the term senile dementia until after autopsies can prove otherwise?
Probably not.
Okay,
Yeah.
so it's either assumed to be Alzheimer's or you're just waiting until a postmortem.
So it could be Lewy body dementia could be vascular dementia, it could be frontotemporal
I
dementia.
see. Okay. And going back to the question about pesticides and other of the pollutants, do you want to name any of the things that are a particular concern, or is that not a stage you're ready to discuss?
Yes, there is no direct correlation in terms of Alzheimer's disease of these pesticides and pollutants like MPTP. They have a correlation with chronic exposure leads to Parkinson's disease. And in Parkinson's disease, not all Parkinson's patients would have dementia. It's majorly a motor disorder. Yeah, so there is not much connection here in Alzheimer's disease. And I mentioned the risk factors here as mostly the lifestyle changes here is governing AD progression.
Any other questions for Dr. Vasta?
This may seem frivolous, but about a month ago, Evergreen Commons had to read the book, see the movie called Still Alice.
Okay.
And the book is fabulous because it's from the patient's point of view, and she is a neurologist.
Mhm.
And it's really a fabulous book to read. A little frightening because you'll say, I do that. Oh my gosh. So I would recommend from a patient's point of view, the book Still Alice.
Definitely, yeah, what's it?
Any other questions?
You said that Van Andel Institute has about 100 brains that were donated. Does Michigan have a brain bank?
Probably this is called the MIND stands for West Michigan neurodegenerative disorders. So that's kind of I think Michigan State University has one.
Pardon?
Michigan University and Michigan
With-
State.
They combined with, yeah, with the three institutions.
Yeah, so probably
30 years ago, yeah.
they have,
That's still going.
yes.
Thank you.
I just wanted to add, I listened to Senator Stabenow recently, and she has a Hope for Alzheimer's Act. I don't know that it has passed in Congress yet, but it is to support families with, or the person with Alzheimer's disease, to set it up that there's a whole care plan for that individual and that they are connected to the appropriate resources.
Yes.
And I think it's something, as we study this topic, that we really need to have our senators in Congress support this.
Yeah.
Any other questions or comments? Well, seeing none, I want to, on behalf of HASP, thank you, Dr. Vasta, for an excellent presentation. Really appreciate it. Give my best wishes to Kate, okay?
Definitely under that.
We really appreciate the VA.